Wait. Good afternoon, everyone. My name is Dayami Lopez. For those don't know me, and I'm part of the rec center. Usually, what we do is we have once a month an idea forum, and there's different people presenting. So this month, I'm very happy to provide two of my students the opportunity to do a presentation here and show some of the data they have collected in the ongoing Alzheimer project. So basically what they do is that they participate in different portions of the project. And the two students is Briaja Michel and Santosh. I always forget his last name. Sorry, Santosh. Briaja actually is no longer a student in my lab because she graduated in December 2023, but she was very happy to come back and show the preliminary data she collected. Normally, undergraduate students have to do 2 semesters of lab module, which is basically doing research in the lab to get an idea of techniques so they can be better prepared for the future when they find a job in the industry. After Briaja finished her presentation, Santosh, who is a master's student in my lab, he's working on his thesis project. Will continue the presentation with some of his own data that he had collected, which is going to be a follow-up for Briaja research. Then the complete team is looking into the effects of inflammation and oxidation on biomarker of Alzheimer's disease. Each of them will have a specific title. The first title is for Briaja, which is the effects of amyloid beta 42 peptide and phospho-tau 3 into 17 and neuraya growth. And I'm gonna let Briaja to take over. So Briaja.
Okay, good afternoon, everyone. So my study was on the effects of amyloid beta 42 and phospho-tau 217 in neurite outgrowth. I'm ready for the next one. Okay, so Alzheimer's disease is a neurodegenerative disease characterized by memory loss, disorientation, the ability to perform routine tasks, and to learn new information. It affects more than 50 million people worldwide, and it's a chance that people 65 or older will have a chance of developing Alzheimer's disease sometime in their life associated with dementia. So biologically, with Alzheimer's disease, it's a very complex disease, and it's thought to have evolved amyloid beta and tau. And these Alzheimer's disease, it gradually damages the brain cells, and it has lesions on cells on the brain, which are extracellular amyloid plaques and intracellular neurofibrillary tangles of hyperphosphorylated tau on cells on the brain, as we can see here. In the first picture with the normal cells of the brain, and then on the person with Alzheimer's disease, they normally form plaques on the cells or around the cells. So what is amyloid beta? Amyloid beta peptides are major components of the plaques that are present in Alzheimer's disease. With this recent studies, it shows that amyloid beta stimulates dysfunction and may play a prominent role in stimulating pro-inflammatory activation in primary micro. Amyloid beta is a protein that results from the cleavage of amyloid precursor protein in a normal integral protein that is located on the plasma membrane. That protein cleaves by B secretase and Y secretase and form amyloid beta peptides, which range in the size from 37 to 49. The monomers are the oligomers, protofibrils, and amyloid beta fibers, and they spread throughout the brain. Ready for the next slide? Okay. So tau and phospho-tau is an impairment of the microtubular binding, which leads to destabilization, and that ultimately leads to the degradation of neurons on the brain cell. And tau that is phosphorylated at the amino acid 217, which appears to discriminate better between individuals with Alzheimer's disease pathology and controls due to large fold change between the groups compared to phospho-tau 181 and phospho-tau 217 become abnormal earlier than it may become abnormal earlier than the phospho-tau 181, but my study was more on phospho-tau 217. And that is a possible marker for Alzheimer's disease. So during the study, I believe that amyloid beta 42 will increase the phosphorylated tau 217 and reduce neural outgrowth in NS1 cells. And in order to do this, we had to determine the effects on varying concentrations of amyloid beta 42 on tau and phospho-tau 217 expression, and to determine the effects of amyloid beta on neural outgrowth and cell viability. So here we use NeuroScreen 1 NS1 cells. We use those because those are the standard model for the neuron system. And I cultured them in RPM 1640 media with 10% FBS and pen strip. This is a photo of the cells under normal conditions. And we use NGF, which is the neurite growth factor, and it promotes growth of neurites, and it will show if the study is really working or not, if the treatment is working or not, because it causes degeneration. So for this experiment, I set 2 different wells. It was a 12-well plate that was treated with amyloid beta 42, NGF, and one section of the well had amyloid beta 42 at 100 nanomolar, then another one will have NGF, and then it will have both amyloid beta and NGF together. And they were treated for 24 hours and 48 hours after sitting in the plate. And after the 48 hours, I collected the media and processed them for Ripple proteins. And then with the 96 well plate, it was an amyloid beta 42 with serial dilution starting at 200 nanomolar in the presence of 100 nanograms per milliliter of NGF. And they had the same process of a 24-hour and 48 hours, but at the end, it was the plate was used in in-cell Western and to count neurite growth. So here is my Western blots that could show that the antibodies were working. And we used tau 5, the antibody, mostly. We could see a little bit. There's a lot of variation between the samples. So we would repeat it to get better consistency. And then this is just with tau on the phospho-tau 217. We'll also repeat this one because the bars are very light, but you can see where tau will fall right around, say about 40. But it will show that they're working. We decided to perform in-cell Western because it would give us the in-cell Western and Western blotting, Western blots besides ELISA, because we get a little more information, and then we could see the high and the low throughput through the both, just to get a difference between what was really going on, so we could actually see what was going on within the cells. So here is my in-cell Western blotting results. We can see that with both the tau 5 and the phospho-tau 217, the results were getting kind of higher with amyloid beta or NGF present, but we would expect them to be a little higher with NGF since that is the neurite growth factor. And then this is an image of the cells when they were stained. So here we can see that the phospho-tau levels increase and the total tau levels decrease with the increasing amount of amyloid beta 42, and that was from the pictures before of the in-cell Western. And then this is another graph of the amyloid beta concentration with the ratio to tau. And this is very consistent, but just higher around 100 nanograms per mil with amyloid beta 42. So around the middle is where it would peak, and then it would go back down a little. And here is our neurite growth and the viability statement. So the neurite growth was determined using the red stain, and we can see that there's some neurite growth going on here. And then the cell viability was determined using the green stain. And these photos are kind of hard to capture, so we can see some neurite growth in some cell that we had good cell viability within the cells during the experiment. And then these are our charts from those images. So in addition to taking those pictures, we created these graphs. In the outgrowth, the neural outgrowth, it doesn't really show a real difference between the conditions. It's really consistent. And then same with the cell viability, they're both pretty consistent. And then I've manually counted some neurites, and the ones with NGF and amyloid beta were always higher, which was kind of expected since they did have NGF within them. So they had higher neurites than all the other plates with just amyloid beta or NGF alone. And these are some images of the cells. And you can see that with neurite, with NGF and amyloid beta 42, we can see more neurite growth than from the control or just amyloid beta. And with NGF, we have some neurite outgrowth, but that's expected. And then here, this is from the in-cell Western blotting results, the amyloid beta compared to the NOS levels. And that contributed to the development of the neurodegeneration in Alzheimer's disease. Okay. And here, so at the end of the experiment, we gathered that treating with amyloid beta 42 and the amyloid beta 42 peptide affects the tau and the phospho-tau 217 levels treated with NGF, as it was shown in the in-cell Western. And the amyloid beta 2 increased the overall ratio of tau and the total tau, which correlates with the reported role of amyloid beta 42 and phospho-tau 217 they have in Alzheimer's disease that we already know. And then amyloid beta 42 peptide did not significantly affect the neuron growth or cell viability, but that we couldn't really tell due to the short time of incubation, which is only 72 hours. And like in a person, we know it would take more than 72 hours for us to actually see the results happening within the body or within the brain. And the INOS levels were not significantly affected either, further supporting the possibility that incubation time was too short. So if we did this experiment for longer, we may have gotten better or more accurate results. And I would like to thank Catherine, Samaria, and all the students and staff at Bright for having me along the way with this experiment.
And at this point, we're going to take any questions for Briaja.
Thanks very much for a very nice presentation.
Thank you.
I'm wondering how you decided the length of time to choose to incubate.
So being that it was only I was only doing the experiment for a semester and I needed to repeat it multiple times, we just did 24 out like to get it done within that week. I did the experiment like every week, repeatedly.
Uh-huh.
So
was it time constraint?
Yes, ma'am.
Okay.
Yeah, they usually don't spend too much time in the lab because they have to take classes. So this is kind of another class that they take, but they actually do research, not just training on the techniques. So they very constrained on how long they can do the experiments for.
Yeah, no, it's a challenge, but it's a good experience, it seems like, and really helps to move things forward. The next question I have was, did you get the results you were expecting?
Yes and no. I was with the neuron outgrowth. I did expect for it to be more growth in the cells with NGF and amyloid beta together. That was expected, but with the viability and outgrowth, that the levels were really consistent. I expected to see like more changes within the levels, but it was really consistent.
And so, what would be your next step here? If you had longer time.
Yes, ma'am, and that would be in Santos's presentation. What we will do moving forward, or how we would get better results.
But what would you have done if you have more time? That's what's the question.
I would do longer incubation times, and like longer incubation times, and let the treatment sit for longer. So maybe that would have more of an effect on the cells than just doing it after 24 hours, not giving them much time to grow with the treatment.
Thank you. Very nice work.
Thank you.
Any other questions? So right now we're gonna let Santosh share his presentation so I can introduce him. Santosh?
Yes, Dr. Lopez. Yeah. Good afternoon, everyone. I'm trying to share my screen here.
While he's sharing his screen, Santosh, he's actually a master's student and has spent a semester and a half, right, Santosh, trying to optimize some of the condition, because even though one of his cell model is similar to the one that Briaja used, He's trying to optimize human cells instead to try to do similar studies. And he's gonna show some of the results that he got and after some of the optimization.
All right. Good afternoon, everyone. My name is Santosh, and today I'm going to present on my topic, which is effect of inflammation and oxidative stress in neuronal cell. My mentor is Dr. Lopez, and the next slide here. So Briaja already explained what the Alzheimer disease is, and I was just trying to show over here like the healthy brain versus the brain with the Alzheimer disease. And you can see over here like in the Alzheimer disease brain, we have hippocampus shrinkage, ventricle enlargement, cerebral cortex shrinkage, and Alzheimer. Okay, so this one here and the next slide. So here I want to highlight the role of oxidation and inflammation on Alzheimer's disease development and worsening, which is also described by Senor et al. So the pro-inflammatory cytokine contribute to the inflammation and resulting in the accumulation of amyloid beta aggregate, tau hyperphosphorylation, and the neuronal cell. So on the right hand side, you can see the picture over here, healthy neuron versus the brain with the neuron with the Alzheimer disease. So here is the pro-inflammatory factor like the interleukin-6, TNF-alpha, which are the key element that contribute to Alzheimer disease. Also, the oxidative stress lead to oxidative damage, impacting cellular structure in the brain. So the both combined effect like the oxidative stress and the inflammation contribute to the neuroinflammation and cellular damage in the Alzheimer disease. Astrocyte and microglia contribute to the inflammation and can exacerbate the progression of Alzheimer disease. Next slide. So my research aim is to investigate the effect of astaxanthin on tau protein expression and phosphorylation, and the other one is investigate tau protein splicing in response to astaxanthin. All right, so the next slide is this is the cell culture module. So you can see over here NS1 cell as described by the Briaja, and also the human cell SK and SS cell line. So used to study Alzheimer disease and its neuro. Okay, so I use the SK and SS because neuroblastoma origin and is valuable in research oxidation and inflammation role in Alzheimer disease. So in this study, we use the retinoic acid 0.25 micromolar to induce N-type. So the neuron has two type of cell, the S-type and N-type. So we use retinoic acid to produce more N-type. And the amount of N-type used in this experiment was determined in the pilot study. All right, next. So here you can see the treatment. We treat like the here is 5 picogram per ml in IL-1 beta, 50 micromolar S2O2, and 100 nanogram per ml amyloid beta 42, which induce Alzheimer disease. Okay, so the control just have retinoic acid with the media, and interleukin beta here is 202, and this is amyloid beta 42. Okay, so this cell is 100,000 cell per well for 48-hour treatment. And you can visually see over here, okay, let's see here. Okay, there are visual difference in the cell viability under treatment with the interleukin 1 beta and S2A2, but not for the amyloid beta 42. However, so from the Briaja also you can see amyloid beta didn't have an effect on the viability of the cell. So the next, you can see over here, the same effect is seen when doubling the number of the cell here. So we use like the 200,000 cell per well for the 48 hours, and control here IL-1 beta, S2O2, and amyloid beta 42 over here. Okay, so the next we did the MTT assay to determine the cell viability. You can see here the cell viability is higher in the control, and then beta amyloid beta 42 and the IL-1 beta. So the less viability in the S2, almost zero in the S2O2. Okay. All right. Okay, so here determine the protein level determined using the in-cell Western. So over here also the PC. Okay, so the PCS control IL-1 beta, S2O2, amyloid beta 42, and PCSK9. This is for the NS1 cell line, and here is the SS SY5Y, which is derived from the SKN-SS cell. Okay, so the PCSK9, 100 nanogram per ml induce inflammation, but also reduce the BACE1, which has essential role in production of amyloid beta. Therefore, it might have dual role in Alzheimer's disease. Next slide. So the why PCSK9 is important. So several research indicate that measuring PCSK9 is important in Alzheimer's research. PCSK9 regulate LDL receptor that impact cholesterol and brain health. High LDL level linked to increase Alzheimer risks. PCSK9 inhibitor used to treat heart disease could also treat a prevention of Alzheimer's disease. And also PCSK9 level could serve as biomarker for Alzheimer's onset or progression. Right, next slide. So the protein level determined by in-cell Western over here, SS SY5Y cell line. So you can see here control amyloid beta 42, PCSK9, and both. So the amyloid beta 42 increased the phosphorylation of tau, whereas PCSK9 prevented it. In the summary, so the culturing conditions have been optimized to measure total tau and the 3 different phospho-tau in the rat and human neuronal cell, promoting inflammation and oxidation in this cell line induced phosphorylation of the tau. Treating with amyloid beta 42 also induced phosphorylation of tau in this cell line, an effect that appears to be counteracted by PCSK9. Additional experiments are needed to determine the pathway of this effect. So the future study, so gonna be like the astaxanthin related to the neuron, which is also described by the Brocklund et al. So the astaxanthin supplement can improve cognitive and psychomotor function, decreases phospholipid hydroperoxidase, and improve erythrocyte antioxidant status in human clinical trial in Japan. Also, astaxanthin can reduce brain cell death and improve the proliferation of neuron stem cell, protect against damage from ischemia, lower blood pressure, delay stroke incident in stroke in prone rat, and demonstrate a neuroprotective effect on the ischemic mice. So in my research, so I'm going to use the purified form of the astaxanthin, which is derived from the algae. Okay, so has better anti-ROS and inflammatory effect in order to in compared to other astaxanthin above here. Like you can see the HPLC concentration is way better than other 3 astaxanthin for the crystalline astaxanthin over here. Slide. So the future study will be primer to be used, measure splicing. Okay, so the schematic show that the primer designed to detect tau 4R and tau 3R in the RT-PCR. The tau 4R sense primer overlaps exon 9 and 10, whereas the tau 3R sense primer overlap exon 9 and 11. The tau 3R 4R reverse primer is common for both isoform. Tau 4R primer said only detect tau 4R, whereas the tau 3R primer said detect only for the tau 3R. And the sequence for the primer are shown in the above schematic over here. The graph on the right side shows that the primer recognized different level of 3R tau over here. And these are the reference we use for the presentation slide. Yeah, that's all.
So any question for Santosh?
So
Yes.
go ahead. I think this is really fascinating work about the inflammation and the effects. And as a clinician, I'm particularly interested in the anti-lipid treatments. I'm wondering, I don't know of any studies that have taken the patients who are being treated with this medication and looking to see, if they already have Alzheimer's and if they've had any, improvements by taking this. But I found that, and it may just be it hasn't come far enough along in terms of the basic science to look at that. But what do you all think about the? I'd like to hear a little bit more about the lowering of the lipids and how that might play a role in reducing Alzheimer's. I really find that fascinating.
Well, probably I better answer that question. Well, the lowering of the lipids, it's controversy right now because if you go down very low levels, some of these drugs, the PCSK9 inhibitor in particular, had caused some patients have almost not detectable levels of LDL. And in those cases, there have been a controversy that it causes memory issues. So it might have a negative effect because even though in the past it was so that neurons, the myelin in the neurons were all made during the fetal stage and they never got regenerated. It looks like it's not the case because if you don't have enough cholesterol, it will affect that. But in terms of PCSK9 itself, if you have too much of PCSK9, which is usually the case in hypercholesterolemic patients, they could end up having Alzheimer's disease. So not just the cholesterol level itself, but also the PCSK9 has a direct effect on Alzheimer's disease. But having too little PCSK9 also have a negative effect. So my lab promotes for having normal levels of PCSK9, which we are trying to establish through clinical trials. We have some data. We just have to run a bigger clinical trial in that. In terms of whether or not there are some studies where they had looked into the inhibitor usage to prevent Alzheimer's disease, and there is some data on that. I guess it will need a longer clinical trial, but there is some data saying that it will help those patients. So it's really interesting. And actually, this could probably increase the ability of doctors to prescribe the inhibitor, because right now what's going on is there's still a major issue. I had a notification from the Familial Hypercholesterolemia Foundation, the FHA Foundation, saying that there's still a large number of patients that don't get prescribed inhibitor, mostly because they can't afford it, and the insurance will not cover because of the expense. You need to be qualified for, and it's not easy. It's not an easy process to be qualified for, but it will be interesting to see more clinical research done on the use of the inhibitor to prevent Alzheimer's disease.
Yes, this is an area we can think about translational work, because I truly think that this is an area that's going to grow, and it's very fascinating line of inquiry. So you're laying the groundwork there with the basic science. So that's so fascinating. Thank you. And
and also to add to that, PCSK9 function not just in hypercholesterolemia, but now they know that it activates the inflammation cascade. So there are downstream inflammatory factors that are activated for PCSK9. So that's why we are so interested in seeing how controlling PCSK9 levels help with different types of diseases, because inflammation is a bigger thing for every disease. Hypercholesterolemia, cardiovascular diseases, diabetes, they're all related to inflammation, Alzheimer's disease. So that's why we looking into all the different downstream regulators of PCSK9 to prevent specific diseases through that.
I agree. It's going to be amazing, I think, over the next decade to see what transpires there. That's really exciting work.
Yes, we are excited. So a lot of my students work on related to PCSK9 and different areas. So because we are most like, we'd like to look into the connection between different diseases. There is no one person that only have a single disease. They have diabetes and they're having Alzheimer's. They have obesity and they're having cancer. So there is a connection between all the diseases, the most common chronic diseases we get. So looking into those connections, that's our purpose. So
it's wonderful. Great. Thank you so much.
Thanks.
I think the inflammation story is so exciting, and it links, not, as you say, not just Alzheimer's, but many, many other aspects, you know.
Yes.
So I think the door is just opening to those treatments.
Yes.
Great.
Thank you. Any other questions? So no more questions? Any comments? So,
well, I'd like to thank your two students, Dayami, for their very lovely presentations and for the work that they're doing in your lab, which is so amazing. And we're really excited to see where these studies will lead. We know that these are not complete studies, and they're just, you know, the early presentations. So we're very much looking forward to a follow-up.
yeah, we're hoping that especially Santosh will have more data in the next couple months because
Lovely.
he had done a lot of preliminary data trying to optimize different conditions and find the right experimental model. So now we can move on into the trying to prevent those effects by the inflammation and the oxidation, and see what other biomarkers are affected in that process.
Well, that would be wonderful. We'll look forward to that update. Sounds really good.
Okay, thank you.
So thanks, everybody, and thanks, Dayami.